Day 12. Let's go. We will start from watching very recent, like 19 hours or 20 hours ago video from Anton. And he's mentioned it that he talking about origin of life, specifically multicellular one. I have not seen that video. I hope Anton doesn't mind if I watch it on stream. Let's go.
Hello InfoPerson, this is Anton, and today we're going to be discussing some of the recent discoveries in regards to the origins of complex life on planet Earth, and specifically a major breakthrough coming from Australia in helping us understand how pretty much everything around us, including me and you, potentially began. And that's because, based on some of the research from the last few decades, a lot of biologists believe that complex life very likely arose as a result of a kind of a symbiosis between bacteria and other complex cells. But the problem was that this was
So is that symbiosis happen like pretty much what happened with our two cells, the mitochondria region, right? Because that's was a symbiosis between bacteria and our cell, I guess. We provide protection, and mitochondria provides energy. We deliver oxygen. Maybe even deeper. I don't know. I'm gonna guess. By the way, probably if you want to watch Interrupted, Pure Science, just go to channel Anton, subscribe, watch his video, is amazing. Here I'm gonna pause it and talk out loud my thoughts.
mostly a theory or a hypothesis, and there was always a kind of a missing link between simple bacteria and much more complex cells, such as eukaryotic cells, that all of us are made out of. And so biologists have never actually seen this in action until basically extremely recently. And so to find some of these answers, we actually have to travel to a location that most of us have probably never heard of. The location in Western Australia that's actually a World Heritage Site. This is known as Gathagudu, a location off the coast of Western Australia that's also sometimes referred to as the Shark Bay. And here this place is very important for one very specific structure that's actually extremely rare on our planet. This location is home to stromatolites, layered mounds of microbial life that basically resemble rocks but are actually living relics of ecosystem from.
Is that only in this place? Like, I knew Australia is weird, right? With all the like animals, but I'm wondering if there are any other places. Very interesting.
Billions of years ago. And though these have been discovered in other locations like near Saudi Arabia, the study about which we've discussed in one of the videos in the description, normally these are actually kind of rare, but are super important for understanding the evolution of complex life. And in essence, these are microbial mats, or basically a collection of different microbes living together, forming these enormous communities. And they're also oxygen factories. In fact, some of the first bubbles of oxygen that filled Earth's early atmosphere most likely came from the ancestors of very similar structures. So in some sense, you could say that we owe our existence to a lot of these somewhat strange looking.
I mean, obviously I didn't call that. I just mentioned oxygen in a different mechanism, but let's go.
Rocks. But the most recent discovery is not really about oxygen or how these microbes breathe, but about genealogy of our greatest microbial ancestor, the genealogy of so-called archaea. Technically, these are not bacteria and not eukaryotic cells, but are something slightly different and possibly something in between. But to understand this research and why it's so important, let's briefly talk about.
Yeah, that stuff in between, I was talking about like not look at this in its categories, but spectrum. In situation like this is actually perfect example when it's useful to look in a spectrum, because it was always gonna be a few outliers on a border that's you don't know in which category to put it.
our cells, eukaryotes. And so here, all plants, animals, fungi, and a lot of single-cell organisms we usually find.
Hold on, let me call. So, this is mitochondria, from my understanding. And these bubbles, I don't think that's ATP, right? Maybe. Not electrons, not the protons. I'm wondering what those bubbles are. Maybe it's not even, oh yeah, it's not mitochondria. That's a ribosome, right? And the nucleus itself. Yes. So we're looking at this part. So ribosomes, the bubbles are ribosomes, and this purple stuff is rough endoplasmic reticulum. Yeah, that's what it is. In that case, what is blue? I don't know. And what is this? Interesting.
In ponds are what's known as eukaryotes. Basically, our cells have
Oh, that's a cool image. That's a cool image. I need to print that.
complex parts, including nucleus.
Yeah, sorry. So this is mitochondria then. So semi-transparent one was mitochondria. These guys.
Different and possibly something in between. But to understand this research and why it's so important, let's briefly talk about our cells, eukaryotes. And so here, all plants, animals, fungi, and a lot of single-cell organisms we usually find in ponds are what's known as eukaryotes. Basically, our cells have complex parts, including nucleus and mitochondria, and a lot of structures that bacteria do not possess. And for many decades, the explanation for this was the idea known as endosymbiosis, or symbiogenesis. Basically, 2 billion years ago, some kind of a simple bacteria, possibly joined with some kind of an archaea, merging into a single cell and becoming a kind of a proto-eukaryote. And so basically, instead of eating the bacteria, the archaea formed a partnership with it, with the bacterium eventually becoming our mitochondria, the powerhouse of the cell that provides a lot of energy.
That's I called it. That's exactly what it is. But explain in normal language. That's how we become, right? And that bacteria become a mitochondria. Called it.
Energy, but also seems to control our bodies in a lot of other ways. You can learn about this in one of the videos in the description. And so not so long ago, less than a decade ago, completely by accident, researchers discovered a potential host of this initial relationship. That's the Asgard archaea you see right here. And a lot of these microbes were actually named after the Norse gods, such as Loki, Odin, and Thor, mostly because they were discovered near a hydrothermal vent referred to as the Loki's Castle. It's actually very close to Norway, so essentially naming in this case made a lot of sense. And what makes them special is that they do carry a lot of eukaryotic signature proteins and a lot of additional genes that bacteria do not have, but our cells seem to contain as well. And so, for example, proteins for
That's so cool. Now I'm looking at these images. Before it was just pretty to me. Now I'm looking and I know this is lipids, hydrophobic head, and antiphobic, antihydrophobic two tails. And this is protein passages. And this lipid, it's a membrane of the cell. Damn, those 12 days was not wasteful. I like it.
moving the cell membranes and transporting materials that we once thought only complex cells could do. But while scientists have known about these
Clip that, by the way. Trying to get used to those keyboards. Clip that.
Asgard archaea for at least a few years now, They essentially are just a little bit too difficult to study in the lab, mostly because they grow incredibly slowly and usually depend on other organisms for survival. Which basically means that a lot of this was still somewhat hypothetical, and there was no actual evidence that any of this could have happened. And so this is where the team of researchers, including the associate professor Brennan Burns, decided to focus on.
So that's the article, right? That's the paper. So we're reading the paper. This is authors, quotations, citations. They making a claim. They create a experiment, right? That's how it works. That's what scientists to be. So cool.
Including the associate professor Brennan Burns, decided to focus on. They spent 5 years nurturing these microbes and essentially tried to grow them inside the lab based on the samples collected from the Shark Bay.
Look at this. This colony, it's not like a region or district, city. It's not even a country. It's not like even a planet. The amount of organisms there, probably like a galaxy. So we took the entire galaxy and then collected some habitants. Will be a proper name for that?
In other words, they tried to recreate all of this in lab conditions in order to find some kind of an evidence for this endosymbiosis hypothesis. And in this new study published in April of 2026, they have now revealed that they were able to successfully enrich a novel species of Asgard archaeon that you can kind of see in this picture. They also named this, okay, let me try this. This is going to be a tough one. Nerearchaeum marumarumae. With the word marumarumae,
Neo Arcade Maru Maru Maru. Maru Maru Maya. Neo Archaic Maru Maru Maya. Clip that. I'm practicing pronunciation with that. That's so cool. So let me understand what happened. They try to basically put together two organisms and see if they form symbiosis, right?
coming from the local tribal language that basically means ancient home, which basically is what they're trying to recreate here. They're trying to recreate the ancient home of complex life. And so this is where things get a little bit interesting. Here, by using extremely
Let's go.
sensitive 3D imaging techniques, something referred to as electron cryotomography, scientists were able to witness something create the ancient home of complex life. And so this is where things extremely sensitive 3D imaging techniques, something referred to as electron cryotomography.
This is cool. So what's this instrument? Microscope columns. So it's making a slice of images on a not on a molecular level, right? But very close to it.
Scientists were able to witness.
This is amazing. The resolution is amazing on the left side. But I don't see clearly those white stuff. In the image they are dark, right?
Something we've never seen before. The first contact between an Asgard archaeon and a bacterium. And specifically these images show us that the two organisms were physically interacting through very tiny thread-like structures referred to as nanotubes.
I mean, take a look at this. On the right side, I see it clearly. On the left side, I don't. Like, really? So it's interacting through the nanotubes. Those are not the same nanotubes that we have in our cells, are they?
Interacting through very tiny thread-like structures referred to as nanotubes. In other words, here we have the first ever evidence for an actual symbiosis between archaea, and specifically Asgard archaea, and bacteria. But here these microbes were not just touching, they were cooperating. And that's because here by using genetic...
Like, how do you know from the picture? Hopefully not, because from a picture...
sequencing, scientists determined that they seem to be sharing nutrients, including vitamins. Specifically here, the archaea produced
By the way, yesterday I looked into what's the difference between vitamins and what's that? And minerals. No, yeah, totally. I have 1,000 tabs open.
things that bacterium needed, and the bacterium returned the favor, which directly reflects the hypothesized partnership that we believe formed the very first complex cells. So it's kind of like the Shakespearean Romeo and Juliet, where two different families of microbes decided to break tradition and live together. Although, wait, in the Shakespearean story, I think they both die. Here, though, they didn't. And so I guess the question is, why exactly does this matter? Well, this
Dude, that background.
didn't. And so I
So cool, right? So C, D, F, those are different protein, I'm assuming. Small GTPSS surface and others. Beige protein, encapsulate shelf. Langin, ARF, fusion. That's so cool. Propylene, FTSZ, CE, TZ, cytoskeleton.
guess the question is, why exactly does this matter? Well, this Australian discovery is a huge deal because it provides the first visual model and actual evidence for how complex life might have started on a primitive Earth. And so even though for a very long time we only had DNA evidence, or basically just some of the genes that we seem to share, now we can physically see the cellular machinery in action. And
Look at this. You gotta be an artist to draw a right side from a left side. Like half of what's drawn I don't even see. Like this stuff. Where is this? Maybe he has the entrance, but you don't see it. Again, this one white dots. This one. Wow.
and how most of this began. And it also confirms that life didn't just evolve through competition.
This is cool. Vitamins go through the protein. This is membrane again. Amino acids go to the protein. And there is two processes that's how you can go through the protein. That require energy and that doesn't require energy. Natrium plus going out. K plus going out. Osmosis balance. This is mitosis, this is meiosis, this is osmosis. Balance and matter. Preparing the song. I want this image. This is cool image.
But in most cases through extremely complex cooperation. And so without this specific microbial marriage, complex life as we know it would have never existed. Although naturally where this happened is of course currently unknown. But based on some of the other studies, we might start making some conclusions based on what's been discovered in a lot of extreme environments on the planet. So this is not the only study on the evolution of life.
This is interesting. So ferrium 2 plus going back and forth. Oh, this is the pump. ATP, that's what's require energy. It use ATP. One of the charge used, and you have only two charges left, and therefore it's called ADP right here, H plus, and it's going out. Very interesting. That's what I learned, like what I'm talking about charges. I think day 7 or something. Sulfide metabolism. Yeah, I need that image. That's so cool.
And the beginning of complex life, with some of the other recent research investigating at least 2 potential places where all of this could have begun, with the 2 most likely locations being either the deep-sea hydrothermal vents or possibly land-based hot springs. So for example, at least one recent study discovered that minerals called iron sulfides inside of these hot springs seem to trigger chemical reactions that produce methanol, one of the main building blocks of life, which obviously provides a kind of a link between geochemistry and early biology. And so here, when it comes to the cradle of complex life on Earth, a lot of research points at some of the most extreme environments with high pressure and high temperature, such as the previously mentioned Loki's Castle, with many of these systems providing all of the essential ingredients for a prebiotic chemistry: heat that drives complex chemical reactions, minerals that are needed for the development, and longevity in a sense that many of these systems can remain active for thousands or even millions of years. But more intriguingly, at least one other study even suggests that many of these locations could have started naturally as a result of asteroid impacts. Or basically, when a very large rock hits the planet, because it creates intense heat and intense pressure, it can actually form hydrothermal systems in the crater that possibly last for thousands of years, which does give molecules plenty of time to organize into complex structures, and once again provides all of the necessary conditions for life to start evolving. But once again, when it comes to these types of explanations, right now this is still very hypothetical. In other words, it's mostly based on mathematical models and theoretical predictions, not actual physical observations. Nevertheless, a lot of this research strengthens the theory that first of all, life seems to be a kind of a natural byproduct of planetary geology and chemistry, and can emerge in dark pressurized depths in various oceans, which is of course great news for astrobiology, because we might be able to find something similar in places like Enceladus and Europa. But it also tells us that the path to complex life is usually paved with collaboration, because these tiny Asgard microbes in Shark Bay remind us that even the smallest partners can eventually completely change the history of the planet. And so in essence, you and I are literally the result of a 2 billion-year-old friendship between 2 very unlikely species, a tiny microbe and some kind of an unusual archaea. And so what you're seeing here is not just some kind of a rock or some kind of a slime. This is literally an ancient home of the ancestors that made our existence possible. And assuming this is a universal phenomenon, something like this might have happened elsewhere. But importantly, we now have actual physical evidence that this is probably how complex life began. But on that note, if you want to find out more about Asgard Archaea or some of the other discoveries from the last few years, check out some of the previous videos in the description. We'll definitely come back and discuss this more in some of the future videos, but until then, thank you for watching, subscribe, come back tomorrow to learn something else, support this channel on Patreon where you can find additional videos, videos without any ads and can DM me directly, or by joining a channel membership that grants you early access. You can also support this channel by buying the Wonderful Person t-shirt in the description below. Stay wonderful, I'll see you tomorrow, and as always, bye-bye.
Right here. Well said, well said. Yeah, we are children of those interesting marriage. Are we aiming to finish 10 questions today and finally get our score? Possibly, right? I don't see why not. 1-1. What is epigenetics? You mentioned it in several times already. Give me your full understanding. Epigenetics, its ability of organism to change instructions during the lifespan of organism. Meaning some genes can be turned on or off using special chemicals or enzymes. Sorry. I'm trolling. Using special chemicals or special mechanism. So the information that's located in DNA could be altered a little bit. Oh, by the way, I didn't turn on the recording. Classic. Let's try it again. Epigenetic, its ability or function of the organism to change DNA instructions in a living creatures, not by mutation or replication, but by turning on and off different genes using either chemical or enzyme or proteins. I don't know which one of them. Okay, let's go. Clean and accurate. Wow. Let me feel the edges. Changing gene instructions without changing the DNA sequence itself. Correct. This is the correct core definition. The DNA letter ATGC stays the same. What changes is whether the gene is readable or not. Turning genes on and off. Correct. Not by mutation or replication. Correct. Mutation changes the text. Epigenetic changes the accessibility to the text. Very good perspective. Using chemicals, enzyme or proteins, not sure what. It's all of the above, but here's the two main mechanisms. DNA methylation, small chemical tag, methyl group, just one carbon and three hydrogens. One carbon and three hydrogens, okay. Attached directly to DNA. When a gene promoter region gets methylized, it's like putting a padlock on the page of the block. Cellular machinery cannot read it. Gene silenced. Histone modification. I heard that word before in some kind of medicine. DNA wraps around. Oh yeah, I remember. Spools like protein called histones. Those spools like proteins. Those are proteins. Yeah. Remember from the chromosome discussion how 2 meters DNA fits in tiny nucleus? Yes, I remember. Histones can be chemically tagged in various ways. Acetylation loosens the wrapping, gene accessible turn on, methylation or deacetylation tightens. Okay, they loosen me, like I cannot comprehend what happened here. Gene packed away turned off. Oh, I see. Think of it as a difference between Titan wouldn't scroll you can read versus an open book lying flat. Okay. What you didn't mention, but probably want to know. Epigenetic is what makes cell types possible. Oh, right. Right. So basically, epigenetics applied to the stem cells. Sorry, communication from the neighbor cells applied to the stem cells by function of epigenetics. So the certain type, so the stem cells would become a certain type. Every cell in your body, yeah, clip that. Every cell in your body has identical DNA, but a liver cell and neuron behave completely differently. Yes, the different type. The difference is epigenetic. Liver cells have liver genes accessible and neurons genes silenced, and vice versa. This is the mechanism behind stem cells. Called it. Clip that. Epigenetic. This is the mechanism behind stem cells differentiation. Call back to question 23, your base class analogy. As the stem cells specialize, epigenetic marks progressively lock down the genes it won't need it. Yeah, makes sense. Environment influences epigenetics. Diet, stress, exercise, toxin, and even social experiences can modify epigenetic marks. Right, because we don't want to adapt just from generation to generation. We want to adapt, aka at real time, at our lifetime as well. So if you need pretty much to move heavy stuff on a daily basis, epigenetic will signal to your DNA, unblock some page of Hercules, and the growth hormone will start flowing and you become like a bodybuilder. That's how it works. Clip that. I feel like clips that it's become so annoying during the stream. I need a button. Bam. That will record timestamp. And then obviously those timestamps will be splitted, analyzed by AI, and cut it into the media chunks. Small media video clips, small video clips. Silence versus this is environment influence. Yes, experience can modify epigenetic marks. The classified study Dutch hunger winter. The classic study Dutch hunger winter of 1944-45, Second World War. Pregnant women who experienced famine had children and even grandchildren with higher rates of obesity. Wow. Cardiovascular disease and metabolic disorders. Traced to epigenetic changes caused by the famine that persist across generation. Very interesting. Programming analogy. What? 0.7? If DNA is source code, epigenetic is file permissions. Same code base, different modules accessible depends on which user cell type is logged in. And those permissions can be modified at runtime by environment events. Core definition correct and concise, missing the specific mechanism. Yeah, I would like histone modification and methalon. But okay, now I think I will remember how epigenetic actually can affect. And the critical role in the cell differentiation, the stem cells type. 57.5, not bad. Question 41, let's go. What is the difference between somatic cell and germ cell? Whoa, that is the second question that I have no idea. The germ instinctively kind of explains something bad, foreign, but I have no idea. I don't know. That's the second question. I don't know. And somatic is some kind of germ that's in symbiosis with us. Somatic cell, every cell in your body that is not involved in reproduction. Every cell in your body that is not involved in reproduction. So skin, muscles, neurons, liver, blood, all somatic. The word comes from Greek soma, body. These cells have 46 chromosomes and divided by mitosis. I just had a song about it. Germ cells, the lineage that produce sperm and eggs. By the way, they divide 2 times and has only half, right? And that's meiosis probably. Mitosis, normal, meiosis for reproduction. These are the only cells that undergo meiosis, exactly, halving to 23 chromosome. They are the only cells whose DNA gets passed to the next generation. Germ. So it's not a germ then. Somatic.
Germ.
It is germ.
Germ.
Interesting. Why does distinction matter enormously? I mean, yes. I know why it's matter, but we'll read it. Somatic says skin cells that's mutation affects only you and you that cell's decision. If you get a mutation in germ cell, this is also why CRISPR ethics debated is so heated. Call back to question 31. Editing somatic cells gene therapy affects only the parent. Editing germ cells or embryos changes are heritable, affects all future descendants. That is fundamentally differently ethical category. Oh, okay. I thought I'm gonna learn something new. And it concerns to your HIV question earlier. HIV integrates into somatic cells, T cells, not germ cells, which is why it isn't inherited genetically. Yeah, I knew that. That makes sense. By the way, can you use epigenetic to turn off HIV in your DNA? I mean, this is such a dumb question, but I'm gonna ask. So, can you use epigenetics to turn off HIV gene in your somatic T cells? That's question number 1. And question number 2, why don't we trigger clean suicide of T cells who has HIV built into the DNA? I forgot the word for that, clean suicide, when cell kill itself. Apoptosis. Interesting. Looks like you passed the excerpt but didn't type a question yet. Yeah. Getting sloppy. Can we call it a side quest? Yeah. Not a strong one, but side quest count 1. Two excellent questions. Both are actual active research strategies. Let me take them in order. Can you epigenetic silence the HIV gene in T cells? This is a real approach called block and lock. Block and lock, buddy. Block and lock. The idea is exactly what you described. Don't remove the HIV DNA, which is nearly impossible once integrated. Just permanently silence it with epigenetic marks so it can never be read. Now my question is, who's doing the research? When did it start? And why it did not start earlier? Because this is kind of obvious question you ask yourself when you just learn about biology on a day 12, right? On a day 12, you're just saying like, hey, why don't we use epigenetic to disable HIV, integrated HIV in our T cells DNA? Huh? Of course. I've been sarcastic. Clip that. We're probably going into the side quest of figuring out when is that research starters. I guess epigenetic. Here's the problem. HIV actually already gets epigenetically silenced in some cells. That's good. That's what creates the latent reservoir after someone starts antiretroviral drugs. Okay, HIV stops actively replicating, but in a small number of T cells. The viral DNA sits quietly in the chromosome, epigenetically suppressed but not permanently. It's sleeping, not dead. Stop the drug and the virus wake up. The epigenetic silencing is reversible. Oh, so that was actually a real treatment. So it started like years ago, like 50s maybe? Because it's not just questioned it. They questioned it, develop the application and actively using it for 50 years. I mean, don't quote me on the 50 years, but... So the block and lock strategy is trying to make that silencing permanent and irreversible, adding stronger epigenetic lock that can be undone. Right, so you don't have to take it daily medication or weekly. You just go do it once and done and done. These are compounds in development, like dihydrocortistatin A, that target a viral protein called TAT, which HIV needs to reactivate from latency. Early results show it can push the virus into deeper silence, but permanent is hard to guarantee when you are talking about decade of patient's life. Interesting. Second question, why not just trigger apoptosis in HIV carrying T cells? This is the other real strategy called shock and kill. Block and lock and shock and kill. Simple answers to my questions. Let's go. And the reason it's hard is authentication. Yeah, you don't know which T cells has it and which don't. You cannot just go with Microsoft and check the whole 2 meters of DNA or meter of DNA of each cell. The latent reservoir cells look identical to normal T cells. HIV is integrated but silenced. No viral proteins are being made. Nothing is displayed on the cell surface. There is no flag saying I'm infected. The cell passes every inspection. Your immune system cannot see it. a drug can target it. It's a sleeper agent with perfect cover. Right. So shock and kill works in 2 steps. Shock. Use a drug called the latency reservoir agent to wake up the silent HIV, forcing the cell to start making viral proteins. Now the cell is visible. Oh, that's so cool. That's so cool. It has a viral protein on its surface. It's basically you going for hunt for the birds and you shock it and you kill it. Kill once visible, the immune system or engineered T cells or antibodies can recognize and destroy it. Or the cell may trigger its own apoptosis. Apoptosis? How to pronounce it? Apoptosis, yeah.
Apoptosis.
Apoptosis. Once it realizes it's producing a viral material. The problem: current latency reversion agents can wake up all latent cells. Even if you kill 99% of the reservoir, the remaining 0.1 is enough to restart the infection if the drugs are stopped. You need to get essentially every single one, and the reservoir is estimated at roughly 1 million cells hidden in 1 billion of normal T cells. The problem, where this connects to CRISP, there is a third approach, use CRISP. Yeah, that's what I thought about that as well. Obviously, anyone would, because we know that CRISP can cut it out to literally cut the HIV DNA out of the host chromosome. Yeah, here we go. This has been, but it's probably so expensive that it's not even worth. This has been demonstrated in animals models. In 2019, researchers used CRISP plus antiretroviral drugs to completely eliminate HIV from some mice. The challenge is delivery, getting CRISP into every single infected T cell in the human body. Yeah. So all 3 strategies you'd intuitively reach for: silence it, block and lock, kill the carriers, shock and kill, and cut it out, CRISP, excisions, are all being pursued. None has worked well enough yet for a cure, but the combination might eventually get there. And one of the most important questions of all this quiz, the question under the number 42. What is the feedback loop, and can you give biological examples? What's the name of it? Homeostasis? Stasis? Homeostasis? Feedback loop, on a basic example, it's pretty much thermostat or feedback loop in organism. It's a system to control certain volume or behavior of the system. In biology, it's called homeostasis. I cannot pronounce that name, but there is a name for that. And the idea is, not the idea, but the way how it works, it's know its goal, and it's know its current situation, and it's know its need to do something to achieve its goal. For example, there is many feedback loops in human body. One of them to maintain the pH level. different pH levels in different part of the organism. Another one is to maintain a temperature. There is many others, and the idea, like not idea, but the how it works. It know its range or certain goal, or what pH level should be, or the temperature. And when body getting too warm, it triggers certain processes to cool it down. When it's too cold, it triggers certain processes, as shiver, for example, to keep it warm. Oh, I butchered it, so I forgot the word. Let's see. Solid and correct. Let me add the one distinction you missed. Thermostat analogica. Know its goal and current situation. Acts to correct. This is called set point and sensor effector loop. Homeostasis as the overarching concept. Homeostasis. pH maintenance, correct example. Temperature regulation, swearing to cool, shivering to warm, correct. Classic negative feedback example. The distinction you need, there are two types of feedback loops. Negative feedback loop, what you describe, the output opposite the deviation, pushing back towards the set point. Temperature too high, sweat, temperature drops, sweat stops. Blood sugar too high, blood sugar, insulin release, sugar absorbed, insulin stops. This is a thermostat. The vast majority of biological feedback loops are negative. They are stabilized. Positive feedback loop, the hormones, the output amplified the deviation, pushing the farther away from the starting point. This sounds dangerous, and it is. Positive feedback loops are rare in biology. Oh, autoimmune system, right? Autoimmune disease, not system, disease. Is that the one? Starting point. This sounds dangerous, and it is. Positive feedback loop are rare in biology and always have hard stop. Example. So it's like nuclear reaction, right? Childbirth contractions. Baby pushes against cervix, nerve signal. Brain releases oxytocin. Is it the oxytocin that everyone talks about? Not everyone talks about it, but there is a documentary about it. Stronger contraction. Baby pushes harder. More oxytocin.
Oxytocin.
Documentary. Yeah, it is the one, oxytocin. Wow. Stronger contraction escalating loop that only stop when the baby is delivered. The event itself is the switch off. Interesting example, very interesting. I would never guess that. Blood clotting. Small cut, platelets stick, they release chemical attracting more platelets, more chemical, more platelets, cascading until the wound is sealed. Oh, okay. Action potential in neurons. Sodium channels open. Positive charge rushes in nearby channels, detects the voltage change they open to. Wave propagates down the neuron. This is why action potentials are all or nothing. Once triggered, positive feedback carries into completion. So this I don't understand. Is this the explaining how the electrical signal travels through one neuron or through the many? Sodium channel open. Positive charge rushes in nearby channels. Detects the voltage change. They open to. Wave propagates down. I need a visualization of this. Here you go. Harvard, yes.
This is a neuron which has four main receive information. The cell body parts. The dendrites receive information. The cell body processes and integrates that information. The axon carries the information along long distances from one part of the neuron to another, and the axon terminal transmits the information to the next cell in the chain. A bundle of axons traveling together is called a nerve. Nerves can be very long as they often need to transmit information over long distances.
Hold on, so many nerves, right?
Neuron to another, and the axon terminal transmits the information to the next cell in the chain. A bundle of axons
traveling together is a nerve.
Traveling together is called a nerve. Nerves can be very long as they often need to transmit information over long distances. As we just saw, the dendrites are the part of the neuron that receives incoming signals.
So that's many, and then to one, or many to many, but this is receive. So this end always receive from other neurons that from basically these parts. I forgot how they name them. Axon terminal. So this one receives from the axon terminals.
Based on the strength of this incoming stimulation, the neuron must decide whether to pass that signal along or not. If the stimulation is strong enough, the signal is transmitted along the entire length of the axon in a phenomenon called an action potential. When this happens,
Oh wow, I just realized I really thought action potential is between neurons. Like basically this image, you see this thumbnail? I thought that's action potential, so between this end and this. But action potential, it's mean like it started from any of these branches and will go all the way to the end to all of them. Now makes sense. So it's not between different cells, but within same cell.
we say the neuron fires. Transmission of a neuronal signal is entirely dependent on the movement of ions or charged particles. Various ions, including sodium, potassium, and chloride, are unequally distributed between the inside and the outside of the cell. The presence and movement of these ions is not only important when a neuron fires, but also at rest. To start, let's think about the positively charged sodium and potassium ions.
This is definitely a side quest. And I think I'm gonna speed up the speed.
When a neuron is not sending a signal, it is considered to be at rest. In a typical neuron in its resting state, the concentration of sodium ions is higher outside the cell than inside. The relative concentration of potassium ions is the opposite, with more ions inside.
Okay, now I remember those. Action potential for every new neuron takes a lot of ATP, and 20% of all our ATP actually use to move those ions from outside of cells into inside, or maybe from inside to outside. I don't remember which direction, but most of those ATP, not most, 20% of ATP that we produce, that's mitochondria produce for us using oxygen that's been delivered to it through the red blood cells, who inject the DNA to be vehicles for oxygen. It also injects the mitochondria and DNA and will never divide again. And it will die in 120 days. All that left in my head after 12 days. So those oxygen been converted into the energy, also the water, also what was that? Something else to use there. CO2, it's a byproduct. The sugar. And the process called aerobic respiration. And I even have a picture. A diagram of that process. So those ATPs produced moved here, or they produce here and use here to then move those ions.
The cell than outside. This ionic separation occurs right at the cell membrane and creates a chemical gradient across the membrane. Because ions are charged particles, we also need to consider their charge when thinking about their distribution across the membrane. At rest, there are more positively charged ions outside the cell relative to the inside. This creates a difference in charge across the membrane, which is called an electron.
Hold on, we're gonna come back to this. Let me reply to message one quickly.
Electrical gradient. Together with the chemical gradient we already mentioned, we refer to this ionic imbalance as the electrochemical gradient. The difference in total charge inside and outside of the cell is called the membrane potential. At rest, when no signals are being transmitted, a neuronal membrane has a resting potential of approximately.
Oh my god, clip that, by the way, the previous one, right? And I just realized I was showing the picture all the time for the mitochondria, but looks like I was wrong for 12 days. This is how it happens, from thinking you're right here to realizing you're right here, like all the way the drop. So that picture that I was showing, it's not how mitochondria make ATPs. It's how neurons create the action potential to be able to fire and use ATP. Whoa! I was so wrong. My sincere apologies. Ruined it. I need to jump on something else really quick. We'll try to finish watching this a little bit later or finish watching it tomorrow. I will do more study offline. See you, my friends. Big discovery today. Very big. Clip it.